You know that simple pill you swallow, maybe for a headache or an infection? It seems so basic, right? But the moment it goes down, it kicks off this incredible, epic journey through your body. So, let's pull back the curtain and see what's really going on.
I mean, it's something we probably don't even think about. We just expect it to work. But here's the thing. Your body isn't just a passive container. Oh no. It actively engages with that drug, putting it through a seriously complex 4-stage obstacle course.
And this whole process, it's got a pretty fancy name, pharmacokinetics. But don't let that word scare you. It just means what the body does to the drug. And we're going to break it all down using a simple acronym, ADME. That stands for absorption, distribution, metabolism, and excretion. This is our map for the whole trip.
All right, first up, let's talk about absorption. This is step 1. Before a drug can even think about doing its job, it has to get into your system. It's got to get into the bloodstream. This is the getting in phase.
So how does a drug actually cross the microscopic border of our cells? Well, it's got a few tricks up its sleeve. Most drugs that are kinda fat-loving or lipid-soluble can just slide right through the cell wall, easy peasy. Others that are more water-loving have to squeeze through tiny little pores. And some, well, they need a special VIP escort. They actually hitch a ride on proteins to get across. It's way more complex than you'd think.
But, and this is a big but, for any pill you swallow, there's a huge hurdle waiting right at the beginning. It's called the first pass effect. I want you to think of it like a mandatory toll booth. After the drug gets absorbed from your gut, it has to go directly to the liver, and the liver, being the body's main security checkpoint, often zaps a big chunk of it before it even has a chance to get to work.
And that right there, that liver toll, perfectly explains this next concept, bioavailability. Basically, it answers the question, okay, after all that, how much of the actual unchanged drug made it into the bloodstream to do its job? Because of that first pass effect, the bioavailability of a pill is pretty much never 100%.
So let's put this head to head. When a doctor gives you a drug through an IV, it goes straight into your vein. It completely bypasses that liver toll booth. Boom, 100% bioavailability. But that pill you swallow, it has to run the gauntlet. And that's why the dose for a pill is often much higher than the dose for an IV injection of the same drug.
All right, the drug made it past the toll booth and into the bloodstream. Success! Now what? Well, it needs to get around, right? It has to travel to where it's actually needed. This brings us to phase 2, distribution. Welcome to the body's superhighway.
And this part is really, really cool. Once it's in your blood, the drug splits into 2 forms. You've got the free form, which is the active part. This is the stuff that can hop off the highway, get into your tissues, and actually start working. But then there's the bound form, where the drug latches onto proteins. When it's bound, it's inactive, kind of just hanging out. It acts like a storage tank, releasing the drug slowly over time.
But hey, not every exit on this highway is open to the public. Your body has some serious high-security zones. The most famous one is the blood-brain barrier. It's like a super tight bouncer for your central nervous system. Only very specific types of drugs, usually small, fat-soluble ones, get past the velvet rope. The placental barrier during pregnancy works in a similar way, but it's not quite as strict, which is exactly why it's so critical to be careful with medications during pregnancy.
So, the drug has gotten in, it's traveled around, it's done its thing, and now it's time for cleanup. The body can't just let it hang around forever. This is metabolism. Stage 3, where the body starts to chemically break down the drug to get it ready for removal.
And guess who's the head of the cleanup crew? Yep, our old friend the liver. Its #1 job here is pretty simple. Take those fat-soluble drugs that like to stick around in your tissues and chemically change them into water-soluble ones. And why does it do that? Because things that dissolve in water are way, way easier for your kidneys to flush out.
The way the liver does this is actually a brilliant 2-phase operation. In phase 1, a bunch of enzymes go to work modifying the drug's structure, kind of like disassembling it. Then, in phase 2, another team of enzymes comes in and tags that broken down drug with a special molecule, basically stamping it with a big, ready-for-disposal label.
Okay, and this is so important. This enzyme system is why your doctor and pharmacist are always asking what other meds you're on. See, some drugs can speed up those liver enzymes. This is called induction. It makes your body break down other drugs way too fast so they don't work. But other drugs can slow the enzymes down. That's inhibition. This can cause other drugs to build up to dangerous, even toxic levels. It's the science behind most drug interactions.
And that brings us to the grand finale of our journey, excretion. The modified, tagged, and now water-soluble drug is ready to be shown the door. It's time to take out the trash.
When it comes to getting rid of this stuff, the kidneys are the absolute MVPs. As all your blood flows through them, they act like an incredibly sophisticated filter. They pull the drug waste out, they actively pump even more of it into what will become urine, and then they grab back anything useful. Remember how the liver made the drug water-soluble? That's to make sure it can't get reabsorbed right here, ensuring it gets flushed right out of the system.
Now, the kidneys are definitely doing most of the work, but they're not the only way out. Some drugs exit through bile and into the GI tract. Those volatile gases used in general anesthesia, you just breathe them out through your lungs. And tiny amounts can even leave your body through sweat, and really importantly for nursing moms, breast milk.
Okay, let's recap. We've seen the drug get absorbed, distributed, metabolized, and finally excreted. The whole ADME shebang. It's a fascinating biological process for sure, but why does understanding this journey actually matter so much in the real world?
Well, it all boils down to one super important concept, the drug's half-life. All that metabolism and excretion work together to determine how long it takes your body to get rid of exactly half the drug in your system. This one number basically dictates everything about how we use medicine.
And that is the whole reason your prescription bottle has a schedule on it. Take one every 12 hours or twice a day. The goal is to reach what we call a steady state. That's the sweet spot where the amount of drug you're taking in equals the amount your body is getting rid of. It keeps the drug level right in that perfect zone where it's effective but not toxic.
This is also why, in a hospital, especially in an emergency, you might get a big first dose. That's a loading dose, designed to get your drug levels up to that effective range super fast. Then you'll switch to smaller, regular maintenance doses just to keep things in that steady state. It's all just a practical application of this epic journey.
And that brings me to a final thought, and it's a pretty exciting one. We've been talking about this journey as if it's the same for everyone, but it's not. Your genetics, your age, your health, it all changes how your specific body handles a drug. So just imagine a future where your prescription isn't just for a standard dose, but one that's perfectly calculated for your body's unique ADME process. That's where medicine is heading, and it's pretty amazing to think about.
So you take a pill, right? And somehow it just knows where to go and what to do. It feels like pure magic, but it's not. Today, we're going to pull back the curtain on the fascinating science of how a drug actually accomplishes its mission once it's inside your body. I mean, how does a simple painkiller know exactly what to do? It seems almost impossibly specific, right? Well, this whole interaction is really a science with two fundamental sides: what your body does to the drug and what the drug does back to your body. So you can think of it like a two-way street, right? On one side, you have what the body does to the drug. That's a field called pharmacokinetics. But today, we're flipping the coin. We're going to focus on the other side of the story, pharmacodynamics. In other words, what the drug does to the body. All right, so here's our mission for today. First, we'll look at a drug's plan of action. Then we'll get into the classic lock and key idea. After that, we'll talk about power versus precision. And finally, we'll tackle the big question: why is everyone so different? Let's get into it. Okay, so first up, a drug's plan of action. You really have to think of a drug as having this whole toolbox, a bunch of different tactics it can use to get the job done. It's definitely not a one-size-fits-all kind of thing. The strategy has to perfectly match the problem it's trying to solve. And some of these tactics are really straightforward. I mean, think about an antacid. It's just a simple chemical action neutralizing stomach acid. But then you get into some incredibly clever stuff, like antibiotics that literally sabotage a bacteria's entire metabolic pathway. And some are even more, aggressive. You've got cancer drugs that use what's called cytotoxic action to just stop those rogue cells from dividing, right in their tracks. But out of all these different tools in the toolbox, there's one tactic that's by far the most common and, honestly, the most sophisticated. And that's acting on these very specific protein structures called receptors. This is where modern medicine gets so precise. So let's zoom in on that, because this is where the magic really happens. And that brings us to probably the most famous analogy in all of pharmacology, the lock and key. It's such a simple concept, but it's absolutely crucial for understanding how these incredibly targeted drugs work with surgical precision. So what are we talking about here? Well, the receptor, that's the lock. It's a very specific protein structure, either on a cell or inside it, and it's basically just waiting for a signal. And the drug, that's our key. It's a molecule that is shaped perfectly to fit into that lock and trigger some kind of biological response. Okay, but here's where things get really interesting. See, it's not enough for the key to just fit the lock. A drug actually needs two things. First is affinity, which is just a fancy way of saying it has to be the right shape to bind to the lock. But the second thing is efficacy, and that's the ability to actually turn the key and make something happen. And believe it or not all keys that fit the lock can actually turn it. And this difference gives us two main types of drugs. You've got your agonists. Think of these as the on switch. They're the master keys. They bind the receptor, they turn it, and they activate an effect. Then you have the antagonists. These are the blockers. They fit perfectly into the lock, but they don't turn it. They just jam it up, preventing any other key from getting in and working. And even these blockers come in a couple of different flavors. First, you have what's called a reversible competitive blocker. The key word there is competitive. It can actually be pushed out of the lock if enough of the real agonist keys show up. It's a numbers game. But then you have the irreversible kind. This one binds so tightly that it just can't be displaced. It basically changes the lock for good. So that leads to a pretty important question. How do we actually measure a drug's success? I mean, we can't just say, oh, it works. We need to be way more specific. And we do that by using two really critical metrics: efficacy, which you can think of as its power, and potency, which is all about its precision. Let's break that down. Efficacy is all about the power of the drug. What's the absolute maximum biological response it can produce? It's the ceiling. How high can it go? Potency, on the other hand, is about precision. It's a measure of the amount of the drug you need to get an effect. So a really potent drug is one that works at a super low dose. And these graphs show this perfectly. Just look at the one on the left. Drug A is way more potent than drug B. Why? Because its curve is shifted to the left, which means you need a much lower dose to get the same response. Now look at the graph on the right. Drug X has a much higher efficacy than drug Y. You can see it right there. Its curve reaches a higher peak. That means it produces a greater maximum response than drug Y ever could, no matter how much of drug Y you use. But here's the thing. A drug's mission doesn't take place in some perfect, sterile lab. It happens inside the human body, which is, well, it's an incredibly complex and variable environment. And that means what works perfectly for one person might not work at all for someone else. Yeah, there are so many factors that can change a drug's effect. I mean, think about it. Your own age, your sex, your genetics, they all play a huge role. Then there's how the drug is given. A pill is going to act differently than an IV. Over time, you can even build up a tolerance. And of course, a huge one is drug combinations. Other medications you're taking can completely change the game. And this idea, this abnormal reaction to a drug because of some unique genetic or enzyme defect, that's really at the heart of a growing field called pharmacogenetics. It's the whole reason why the exact same medicine at the exact same dose can be a lifesaver for one person and totally ineffective, or maybe even harmful for somebody else. It really does come down to our unique, individual genetic makeup. And speaking of combining drugs, they almost never just add up neatly. Sometimes they team up to create a kind of super effect. That's called synergism, and it's where 1 + 1 equals way more than 2. But on the flip side, they can also work against each other, basically fighting it out and reducing the overall effect. And that's called antagonism. So, knowing all this, what question will you ask your doctor about how your next prescription is designed to work? Because ultimately, understanding these core principles, from a drug's plan of action to the lock and key to all the variables in our own bodies, it really empowers you. It lets you have a much more informed conversation about your own health and your treatment. It takes medicine out of the realm of magic and turns it into a science that you can actually understand and engage with.
You've probably taken an antibiotic at some point in your life, right? But if you ever stop to wonder what's actually happening inside your body, it's not just some magic pill. It's a highly targeted, microscopic military campaign. So today, let's dive into the 4 brilliant strategies these drugs use to take down bacteria from the inside out.
First things first, you need to stop thinking of a bacterium as just some simple little germ. Instead, I want you to picture a microscopic fortress. Seriously, it's got everything: high-tech defensive walls, heavily guarded gates controlling supplies, internal factories churning out essential parts, and even a command center with all the master blueprints. our drugs are designed to exploit specific weaknesses in this fortress.
So you've got this tiny fortress setting up shop inside you. How in the world do you fight back? And here's the real trick. How do you take out that fortress without causing a ton of collateral damage to your own cells? Well, that is the genius behind how these antimicrobial drugs work.
And it turns out there's a 4-point battle plan. We're going to break down each one. First, a direct assault on the walls. Second, a bit of sabotage at the gates. Third, a mission to shut down the internal factories. And finally, a top-secret operation to corrupt their master blueprints.
All right, first up, let's talk strategy 1. We're targeting the cell wall. This is the most direct approach.
So what is this cell wall? Well, think of it as the literal stone walls of a castle. It's a tough, rigid layer that gives the bacteria its shape and, more importantly, stops it from bursting under pressure. But here is the absolute key, the brilliant part. Our cells, we don't have them. This makes the cell wall a perfect target. We can launch an attack that only harms the invaders.
Now, there are a couple of really smart ways to bring this wall down. See, the wall is built with these molecular bricks. Some drugs, like vancomycin, are designed to stop the workers from even laying the bricks down in the first place. But others, like the famous penicillin family, let the bricks get laid, but they sabotage the mortar that holds them all together. Either way, no mortar, the wall just falls apart. It's a guaranteed collapse.
And this wall-busting strategy is behind some of the most famous antibiotics out there. I mean, you've definitely heard of penicillins. They're the ones that mess with the mortar. Then you've got drugs like vancomycin, often a heavy hitter saved for tougher infections. And it's a perfect example of a drug that stops construction right at the bricklaying stage. Okay, so a direct assault is one way, but what about strategy 2? Well, now we're going to get a little sneakier and target the cell membrane.
Just inside that main wall is the cell membrane. This isn't really a wall, it's more like a bouncer, a super strict gatekeeper that controls literally everything that goes in and everything that comes out. So what do our drugs do? They essentially punch holes in it. They create leaks. And when all the important stuff inside the cell starts leaking out, well, it's game over for the bacterium, and it happens fast.
A great example here are the polymyxins. You can really think of them as saboteurs. Their whole mission is just to poke holes in that gatekeeper membrane, spring a bunch of leaks, and let the whole fortress collapse on itself from the inside.
All right, on to our third line of attack. We're moving from the outer defenses to the inside. This is all about industrial sabotage. We're going after the protein factories.
You know, every living cell needs proteins to function, and they're built in these tiny factories called ribosomes. And here's that clever trick again. It turns out bacterial ribosomes are built differently than ours. They're like a different model. This means we can design drugs that specifically jam up their assembly lines while our own factories keep running just fine.
And we can get incredibly precise with this. The factory, the ribosome, is made of two main parts. Some drugs, like tetracyclines, go for the smaller part. They basically block the loading dock so the raw materials can't even get in. But other drugs, like macrolides, target the larger part, jamming up the end of the assembly line so the finished product can't get out. The end result is the same. Production just grinds to a halt. This factory shutdown strategy is incredibly effective, and it's used by some really common antibiotics, like tetracyclines and erythromycin. They're all industrial saboteurs. Their entire job is to sneak inside that fortress and shut down the enemy's production lines for good.
Finally, this brings us to our fourth and, you could say, our most sophisticated strategy of all. Now we're going for the command and control center. We're targeting the genetic code itself. Look, every single instruction the bacterium needs to live, to build new parts, and to replicate, it's all written down in its DNA and RNA. These are the master blueprints. So if you can mess with those blueprints, if you can shred them or lock them away or just make them unreadable, the entire operation stops cold. A fantastic example of this is a drug called cotrimoxazole. Now get this, it's actually two different drugs working as a team. See, to make new DNA blueprints, bacteria need something called folic acid. The first drug blocks one step of the folic acid production line. Then the second drug blocks the very next step. It's this incredible 1-2 punch that completely shuts down production. It's just devastatingly effective.
But that's not the only way to correct the blueprints. Other drugs, like quinolones, do something different. They prevent the DNA from uncoiling so it can be copied. It's like locking the blueprints away in a safe and throwing away the key. Then you have others, like rifampicin, which go after the enzyme that reads the blueprints. So even if the safe is open, no one can actually read the instructions.
So there you have it, the antimicrobial arsenal. 4 distinct lines of attack: blowing up the walls, sabotaging the gates, shutting down the factories, and corrupting the blueprints. Having all these different options is absolutely crucial.
And it all comes back to this one core principle: selective toxicity. It's a fancy way of saying the drug is toxic to them, but not to us. Every single strategy we just talked about works because it exploits a fundamental difference between their cells and our cells. It's what makes this a targeted strike, not just carpet bombing.
But, and this is a huge but, we're not fighting a static enemy. This is a full-blown arms race. For every attack we develop, bacteria are working around the clock to evolve new defenses. They develop little shields that destroy our drugs or tiny pumps that just spit them right back out. This is antibiotic resistance, and it's exactly why our diverse arsenal is so critical. When they figure out how to defend against one attack, we have to be ready with another one.
And this leaves us with a final crucial question. The battle is never truly over. As bacteria constantly evolve to counter our best drugs, the challenge for science is to find new weaknesses in their defenses and to design the next generation of superweapons for our antimicrobial arsenal.